Hi everyone, so now we're on to the peripheral nervous system. If you haven't done so, make sure you watch my central nervous system lecture video. Now we're going to be talking about the peripheral nervous system, and remember peripheral means off to the side, so that includes all of the nerves and the ganglion, everything except for the brain and the spinal cord. When you look at the peripheral nervous system, there are two general pathways. You have sensory pathways that bring sensory information towards the central nervous system. They're also known as afferent or afferent pathways, brings it towards the central nervous system. And then motor pathways, also known as efferent pathways or efferent pathways, bring response information away from the central nervous system and towards effector cells such as skeletal muscle or cardiac muscle or glands, things like that. This lecture we're going to be focusing on sensory pathways. Let's start with looking at the different types of sensory receptors. Receptors are excitable, which means that they're able to create electricity. And they're going to start with graded potentials. And these sensory receptors can either be neurons themselves, such as the touch receptors that are found in the skin. You may be familiar with Physi1 and corpuscle from anatomy or any of those free nerve endings. These are the dendritic ends of sensory neurons. Or sometimes those sensory receptors are other cells or modified epithelial cells that then communicate or synapse with a sensory neuron. So for example, in the ears, you have the receptor cell called hair cells, and they are modified epithelial cells that have the ability to create graded potentials and release neurotransmitters that then synapse or connect to the sensory neuron. Okay, so let's look at those different types of receptors. They can be classified or put into categories based on the function. And if we're talking about the function of the sensory receptors, what we're really talking about is what type of stimulus do they respond to. So these words are pretty straightforward. They tell you exactly what they are. Chemoreceptors respond to chemicals, so chemicals. Photoreceptors respond to photons of light, okay, so light. Thermoreceptors respond to thermal changes or temperature changes. Usually students get those three pretty easily because they kind of just make sense and are right in the name. Now the mechanoreceptors sometimes can be a bit more challenging. What mechanoreceptors respond to is mechanical changes, physical changes. And if you're thinking about what that means for a cell, what we're really talking about is any time you have a change in the shape of the cell. So for example, these hair cells right here that are found in the ear, they respond, they create electricity when their little hairs, which are called stereocilia, on the top of them bend. So any kind of bending or stretching, like you may have seen in the stretch reflexes, or touch or pressure, anytime you would cause any physical change in the shape of the cell that depolarizes it or activates it, that is called a mechanoreceptor. So all of the different types of receptors found in the body, they can fit into one of these four functional classifications. You can also classify receptors based on the location. So these are broader categories and include examples of the functional classifications here. So for example, you can have exteroreceptors, extero as in external. Anytime you have receptors that are found on the outside of your body, such as any of the touch or cutaneous receptors found in your skin, or your special senses. Your special senses include things like taste and smell and hearing and vision. Anything that responds to external stimulus, so outside of the body, those all could be grouped together as exteroreceptors. Now, please note again, this is a very broad category because those exteroreceptors can these exteroceptors include more specific categories of, for example, touch receptors. The touch receptors would be an example of a mechanoreceptor, and things like sense of smell, those again would be chemoreceptors. Okay, so this exteroceptors is a broader category than the functional classifications. So exteroceptors, external stimuli. Interoceptors would be internal stimuli, right? So what this is information that is generated inside of the body. So for example, blood pressure. Blood pressure is going to be monitored using mechanoreceptors that regulate the amount of blood vessel stretch. And so as you increase blood pressure, the blood vessels, the walls of the blood vessels would be stretching outward. And that's how your body knows that the blood pressure has increased. So because this is information from the inside of your body, that would be an interoceptor, internal stimulus. Now there's a third category, and the third category is sort of all kind of out to itself. Those are called proprioceptors, and the proprioceptors monitor specifically body positioning and coordinating movements. Okay, so for example, here you have your bone, there's your muscle. The structures that connect the muscle to the bone, those are called tendons. Running through these tendons, you have stretch receptors, and if there's stretch receptors, what would be their functional classification? Okay, so remember, anytime you have stretching or bending or anything like that, that would be a mechanoreceptor. Okay, so you've got mechanoreceptors embedded in that tendon, and their job is to monitor any stretch by that muscle, right? So when the muscle contracts, it will stretch the tendon, and this tendon will provide feedback to the central nervous system, and that helps the cerebellum smooth out and coordinate those movements. That's called the Golgi tendon. Right, so because this provides information about what the body is doing and how the body is moving, that would be classified as a proprioceptor. Okay, another term that you should be familiar with is nociceptors. Noci means harm, and what this is a broad category of receptors that specifically create the sensation of pain. So they're pain receptors. Nociceptors are pain receptors. And they're going to be responding to a wide variety of different types of stimulus, because lots of different things can hurt, right? So for example, extreme temperature, either too hot or too cold, or extreme chemicals such as acids on the skin, or extreme pressure right here with this hammer. So there's different types of receptors, but they are bringing pain information to the brain. And so they're all grouped together and classified as nociceptors. So from here on out, just think of nociceptors as pain receptors. Okay, so now let's talk about, we're going to get into something that you're going to be doing in lab. We're going to be trying to measure receptive fields. So what receptive field is the area of the skin that is covered by one single neuron, one single receptor. So for example, Physi1 corpuscles monitor deep pression. Those are the ones that look like onions with lots and lots of layers. They monitor deep pressure, and they're not very dense, so you don't have very many Physi1 corpuscles in your skin, and so those areas will cover lots of area of the skin. So for example, if you look at this picture right here, the black dot is supposed to represent the location of that one receptor, and the blue area represents the area that will activate that one single Physi1 corpuscle. Okay, so for example, if I have deep pressure here, or here, or here, any of those places would activate this one single receptor. So this area is called the receptive field for that receptor. So that's the receptive field. Now, please notice that the larger the receptive field, the less dense the number of receptors are, because they have to cover larger areas because there's fewer of them. Okay, so to illustrate that, let's look at Messener's corpuscles. And Messener's corpuscles, they are responsible for light or fine touch, sort of like a gentle sensation on the skin. And they are especially located in, like, for example, the fingertips. So if you look at those fingertips, you see all these black dots. Those are the receptive fields of those Messener's corpuscles. Because there's a whole bunch of them, lots of them packed into it, so a higher density of Messener's corpuscles in that area, the receptive field for these types of receptors is going to be much smaller than the receptive field of Physi1 corpuscles. Okay, so just understand the relationship between density and receptive field. It's an inverse relationship. Low density, bigger receptive field, high density, lower receptive field. So the way that you can approximate or try to measure the receptive field of a receptor, the receptive field of a receptor is the 2-point touch threshold. And you're going to be doing this in lab. This is your nervous system lab. So like, I think it was 7C or something like that. And what you're going to be doing is you're going to have 2 points. You'll start with the 2 points close together, and then you'll slowly increase the distance between the points and see at what point does the subject feel 2 touches instead of 1 single touch. Okay, so you'll start with them closer together, and at the very beginning, it'll feel like they're only being touched with 1 single point, and that's because only 1 single receptor is covering that area, so it can only feel like 1 point. But once you get it further apart, such that you activate more than 1 sensory neuron, now that the subject will be able to feel the 2 separate points. So you'll measure the distance across for those 2 separate points, and that'll tell you the 2-point touch threshold for that area. Okay, so you're going to be measuring multiple different parts of the body and trying to see, well, where are the receptors more dense than others? And remember that smaller receptive fields mean more dense, and bigger receptive fields mean less dense. And that's going to translate to the area that is covered in the brain. So remember, we talked about the cerebral cortex has functional regions that cover what particular type of sensation. So in the very top, okay, so at the very top of your head, right here, you have what's called the somatosensory cortex area. Somato means kind of body-wide, so body-wide sensation. These are all of the general sensations that you can receive through your skin. So information from all of these touch receptors are going to travel to this one specific part of the brain. And the thing that I find very interesting is that the amount of space that area takes up is not proportional to the amount of body space it takes up. Okay, so for example, your lips and your tongue have a very, very high density of receptors, and so they're going to have much bigger areas on the somatosensory cortex than areas, for example, like the back of the leg, where you have very few receptors. The very few receptors is going to have a much smaller area within the somatosensory cortex. Okay, and this just has to do with the number of receptors, right? If you have receptors coming in, you'll need more places for them to synapse, and therefore it makes sense that they would take up more space in the cortex. Okay, so higher density means more space devoted to that particular body part. Okay, now let's talk about what happens after you create that stimulus. And I've talked about this already with the neuron lecture, but let's revisit some of the main points. After you have a stimulus, remember the stimulus is going to be appropriate to that particular type of receptor. Once you have a stimulus, that's going to create a graded potential, okay? And those graded potential, graded means it can vary in the charge. Those graded potentials can either be depolarizing, becoming more positive, or hyperpolarizing, becoming more negative. And again, which direction it goes depends entirely upon the type of stimulus and the type of receptor. Okay, so you have depolarization becoming more positive, hyperpolarization becoming more negative. Okay, so when you think about how it proceeds from there, it really depends on whether or not you're talking about a one-cell system. And what I mean by one-cell system is that the sensory neuron itself is the receptor, or if it's a two-cell system where you have a separate modified epithelial cell that then talks to or synapses with a sensory neuron. If you have a one-cell system, okay, so for example, those free nerve endings that are found in the touch receptors of your skin, that means that the sensory neuron is the one that received the stimulus in the first place. Okay, so if it is a depolarizing stimulus, again, depolarizing means becoming more positive, those depolarizations can summate or do summations, which means that they can stack on top of each other to increase the amount of positive charge. And if they are strong enough or stacked on top of enough to reach the threshold, that's then going to trigger an action potential once it reaches the axon terminal, the axon helix, which is the very initial segment of the axon. Okay, so those depolarizations will spread outwards and stack on top of each other until they reach the axon hillock. And if it's strong enough to reach the threshold, that'll create an action potential. And then those action potentials will be propagated or conducted along the axon towards the central nervous system. Okay, so remember, depolarizations add up, hyperpolarizations subtract. Okay, so if they get the hyperpolarization, that brings them further away from the threshold. And if they're further away from the threshold, that means that they cannot create action potentials and therefore they will not bring information towards the brain. Okay, so that is called the generator potential, which just means one single sensory neuron brings the information. It does it all. The other type is called a receptor potential, and this is the two-cell system, which means that you have a modified epithelial cell doing the actually receiving the sensory input, and then it has to synapse with a sensory neuron in order to take it to the brain. And so this works because that sensory receptor, okay, so for example, the hair receptors in the ears, they have the ability to release neurotransmitters. And so when you have a depolarization, so you have a stimulus creating a graded depolarization, what that's going to do is that's going to trigger the release of neurotransmitters. And those neurotransmitters would then bind to receptors on the sensory neuron. And when they bind to the receptors on the sensory neuron, that's going to create a graded potential in the sensory neuron, which will lead to an action potential, and then that'll be conducted all the way to the brain, right? So if you have hyperpolarization, for example, this is how the photoreceptors in the eyes actually work. If you have hyperpolarization, what that does is, remember, you are further away, you're more negative. That'll result in less neurotransmitter being released. Less neurotransmitter means that the graded potential on the sensory neuron is further away from threshold. Further away from threshold means no action potential. Okay, so depolarization means more neurotransmitter, more action potential, more information for the brain. Hyperpolarization means less neurotransmitter, no action potential, no information going to the brain. Okay, so that's why I try to choose depolarization, green means go, hyperpolarization, red means stop. Okay, so let's talk about decoding the strength of the stimulus. So remember that action potentials are all or nothing. Okay, so what that means is once you reach that threshold, okay, whatever that threshold is, once you reach it, it's going to fire exactly the same way every time. Okay, so if you look at this peak right here, the amplitude or the amount of change in voltage is always the same. So there's no summations and it does not vary in voltage. Okay, it is not graded. Okay, so it'll be exactly the same way. So the only way that you can tell differences in the strength of the stimulus is how often action potentials are generated, right? So if you go back here, remember we have neurotransmitter, they have graded potential. So if you have a strong stimulus, you're going to make more action potentials. And if you look at this right here, here is the stimulus. And these peaks show you the strength of the stimulus. So very, very strong stimulus creates more action potentials, and the frequency of the action potentials that travel to the brain are going to be decoded by your brain as stronger stimulus. We've talked about that already before in the neuron lecture. Okay, that leads us to an idea of sensory adaptation. What does it mean to adapt? Okay, so think about adapt. Adapt means to change, or you can think of it as kind of getting used to the current situation. So when you have continuous stimulus, for example, when you put your watch on, at first that watch, you're going to feel it, right? You have perception of the watch. You think, oh, yep, my watch is on. You can feel your watch. But as you go throughout your day, you're going to feel, you're going to lose perception of it. Okay, you won't feel the watch and you won't be feeling the touch receptors on that watch. Okay, so this would be an example of sensory adaptation. You lose the perception of the watch. Now, thinking about as an organism, why would it be advantageous to lose perception of a stimulus? What do you think? Okay, so really want to think about this. If your brain is trying to keep you alive, what it really needs to worry about are changes in your environment, right? You don't need to worry about every single stimulus, because if it's constantly the same, it's not going to affect or alter your homeostasis, right? So it's more important that your brain is focusing on the things that are changing and not so much the things that are not changing. So sensory adaptation allows you to free up some of the processing speed in your brain so that you can focus and prioritize changes in your environment instead of things that are staying the same. Now, one thing I did want to point out is that your nociceptors, your pain receptors, do not adapt. Why do you think that would be important? Okay, so remember pain. Pain means harm. You don't want to adapt to harm. You need to make sure that you address the harm and get away from the harm, right? So this nociceptors continue to produce the sensation of pain until you get away from the thing that's causing the pain. There are two different types of receptors, or two categories of receptors. You have tonic receptors and phasic receptors. Tonic receptors means that they are slow to adapt, or sometimes they do not adapt at all. And so what that means is if you continue the stimulus, okay, so for example, you shock it for a long time, or you press on something for a long time, or whatever it is, pain, maybe push on it really hard for a really long time, it will create the action potentials for the entire duration or the whole length of the stimulus, right? So if you have the stimulus for the entire length, it'll create action potentials the entire time. That means that it is non-adapting. or sometimes very slow adapting. So an example of atonic receptors would be those nociceptors that do not adapt. You get pain sensation the entire time of the stimulus. The other type is phasic receptors. And phasic receptors create action potentials at the beginning of the stimulus, but with continued stimulus, they adapt so that you're not creating any more action potentials, and no more action potentials means you're not bringing the information to the central nervous system, so your awareness or your perception of that sensation goes away. Those are fast adapting or phasic receptors. Those would be things like your thermoreceptors are phasic receptors, or your touch receptors are phasic receptors. They create a signal at first, but then they stop creating a signal as the stimulus continues. Okay, so let's just generally talk about how we bring the information to the brain. You have a sensory neuron that'll create action potentials that run to the central nervous system. The spinal cord, I should say, go to the spinal cord. From there, the interneurons will bring that information up through the thalamus, through the pons, and then the thalamus is going to reroute the information to the appropriate region of the cerebral cortex. Then that's when you have perception or awareness of that sensation. Okay, that's enough about sensory pathways. You can move on to the motor pathways. See you later.